Okay, welcome to our last recording for our genetics unit. This is going to be all about genetic disorders. So we're going to talk about what they are, different mutations that cause them, and some very specific examples of genetic disorders before we get into your research project, which is all about various genetic disorders. So what is a genetic disorder? Well, it's a disease that's caused by an abnormality in an individual's DNA. So that just means that something changed in the DNA and it led to a negative consequence, right? It led to a disease. Now there's different types here. There's a single gene disorder like point mutations or additions and deletions. Chromosomal abnormalities where either an entire chromosome is missing or added, or a large portion of a single chromosome is either missing or added. And then the third type that we'll discuss is multifactorial disorders, which just means that there's a lot of different things that contribute to the disorder. So it might be multiple gene mutations or gene mutations and environmental factors, or any combination of that and additional factors. So talking about single gene mutations, this was that first kind of branch of types of mutations. They're just changes in a DNA sequence. And just as like a little refresher, DNA sequences are made up of 4 nucleotides, A, T, C, and G. Now the mutations are changes within those nucleotides, whether it's a single nucleotide like just an A, right, just adenine, or the amino acids that those nucleotides are coding for. So mutations change the order of those nucleotides. If you have a different order, that means you have a different amino acid, right? If you change the order of the nucleotides, you're changing what it's coding for. If you change what it's coding for, then you're changing the protein. And if you change the protein structure, you're changing the protein function. So if you mess with the structure of it, or the nucleotides that make it up, you're going to change what that does. And it's not always a good thing. Case in point, these genetic disorders, right? So some consequences here is usually it results in either the absence of a protein or the production of an altered form of the protein, meaning that it's not doing what you want it to do. Most single gene disorders are recessive because at least one of your alleles is going to produce the functional one. So that functional allele is going to mask the allele that holds that gene disorder. And we saw this in our adventure in dog hair, right? If you had even one of the capital letters, you were getting a functional protein instead of a non-functional allele or a non-functional protein. So now let's talk about very specific types of mutations. So there's 6 total that we're going to talk about. The first one is point mutations. So point mutations are just where one singular base pair changes, right? So like one single nucleotide changes, but because of that one nucleotide change, one single amino acid changes, right? So that's what it's coding for. Frameshift mutations are like insertions or deletions of one or more of the base pairs. So again, base pairs is synonymous with nucleotides. So if you change and delete or insert a single base pair somewhere, that means everything that comes after it is either moved up one or moved back one, right? Because you've inserted an extra letter or deleted a single letter out of the code that you had. So it means that all of the amino acids after that mutation have changed, and it completely changes the protein. It makes it something totally, totally different. Deletion, of course, is deletion of part of a chromosome. So you can take like a chunk out of a chromosome and then all that genetic material is just gone. The last 3 are a little bit more complicated. So translocation is the transfer of one segment of a chromosome to another. So that could be like in this picture, a section of chromosome 4 being translocated onto chromosome 20. So that's a huge jump, right? Chromosome 4 is much larger than chromosome 20. So now chromosome 20 has like all these new genes, but it doesn't make sense because your body doesn't read those genes in the same way as they would on chromosome 4 because your body is set up to read chromosomes where they should be. Inversion is the reversal of a section of a chromosome. And you can see this in that picture again on the bottom. You can break a chromosome in a certain area and then basically like flip it upside down and then put it back in. And again, this is going to mess with the reading of the amino acids and reading of the nucleotides because you're changing the order of the code. And then finally, aneuploidy is the gain or loss of an entire chromosome due to non-disjunction. Non-disjunction, you can like underline that, that's a vocab word. Non-disjunction we're going to talk way more about on the next couple of slides, but the process of non-disjunction happening leads to aneuploidy mutations. So what is non-disjunction? If you remember freshman biology, you've definitely heard this word before. What it is when homologous chromosomes don't separate properly or correctly during meiosis. So you're supposed to end up with an equal amount of chromosomes in each of your gametes, right? In each of your sperm and egg cells, you should have the same amount of chromosomes. What non-disjunction means is that they don't separate properly, which means that in some of them, you're going to have an extra one, and in some of them, you're going to be missing one. So this is just showing meiosis. We've sort of talked about it, but again, not in like any crazy detail, but non-disjunction can happen in two different spots in meiosis. During meiosis 1, it could happen when you have your homologous chromosomes and then they split and then all of your gametes are messed up, right? All of your gametes have the wrong amount of chromatids in them. Or it can happen in meiosis 2, right? This is the same phase of meiosis that it's happening, anaphase, but instead of separating in meiosis 1, it doesn't separate in meiosis 2. And in this case, half of your gametes are going to be fine, and half of your gametes are going to have an incorrect number of chromosomes. So if non-disjunction happens in meiosis 2, only half of your gametes are affected. If non-disjunction happens in meiosis 1, all of your gametes are affected. Non-disjunction can also happen with sex chromosomes, and if it happens with sex chromosomes, it is always going to lead to a genetic disorder. So if you have female non-disjunction, where you end up with two X's in one gamete and no X's in another, that can lead to triple X syndrome or Turner syndrome. And if you're matching up with your sperm, for males, it can lead to Klinefelter syndrome when you have an extra X, so XXY, or if you have no X and just a Y, it's non-viable and you're not going to be able to have an offspring with that genome. So now that was kind of like the basics of genetic mutations, non-disjunction, just a general overview, and now we're going to start talking more specifically about disorders. None of the disorders that I'm talking about are going to be options for you to do your research project on, but this is just good examples, and these examples will be on your test. So our first one is a single gene disorder. An example of this is sickle cell anemia. This primarily affects people from Africa. It's a recessive inheritance pattern, so that means that you have to have two recessive alleles to show this disorder. It has a relatively high rate of relevance, rate of inheritance. 1 out of 400 African Americans have sickle cell anemia. What this is a change in the shape of red blood cells. So on the left here, this is a normal red blood cell. And on the right here, this is a sickle-celled red blood cell. And you can see that it does not look the same, and it does not look like it can do its job right. So sickle cells, they can't carry oxygen, and because they're so oddly shaped, they don't fit well between your arteries and veins, so they get stuck on each other very frequently, so not great. Being heterozygous means that you actually show a little bit of both, so you have normal red blood cells and you have a little bit of sickle cell. It doesn't immediately cancel out sickle cell anemia if you are heterozygous, which is interesting. The cause of sickle cell anemia, like I said, it's a single gene disorder, so it actually only affects one singular amino acid. It is a point mutation. It is one single nucleotide changes, and it leads to one change in the amino acid, and it leads to this disorder. Another single gene disorder is cystic fibrosis. This primarily affects white people of European descent. It's about 1 in every 2,500 births, and 1 in 25 is a carrier, so that's pretty high. What cystic fibrosis is all about is the ability to have a functioning protein that moves chloride, or like salt, in your body across cell membranes. So if you have a normal allele, that means that you can move salt across cell membranes. When you move salt across membranes, you also are going to bring water with you, because water is going to move to maintain homeostasis, or maintain an equilibrium in your body. If you have this disorder, though, then you have, it limits the movement of this chloride, it limits the movement of this salt, which means it also limits the movement of water across the cell membrane, which means that your mucus is not as watery because water's not moving. So it's really thick and sticky mucus all over your lungs and your sweat glands. And so mucus builds up in these areas, including the pancreas, lungs, digestive tract, and it causes a lot of bacterial infections. And so if you do not get treatment for this, children will die before 5 years old, just because this buildup makes it very, very difficult to breathe and maintain any sort of balance within your body. But with treatment, you can live past their late 20s. Nowadays, you can live even longer than that because the treatment has improved. So this is just kind of explaining a little bit more what I was talking about. So in normal lungs, if chloride moves, then water is going to move with it. In cystic fibrosis, though, chloride does not move, so water also doesn't move. And so because chloride isn't moving and water isn't being moved through the cell membrane, all of this mucus on these mucus secreting glands are not nice and watery and kind of like thin like they should be in a normal functioning lung, and instead they look like this, where you have a lot of bacteria and mucus buildup, and it's really thick, and it blocks off a lot of your glands, and you can't secrete stuff easily, and it just makes it really, really difficult for your normal cells to function in the way that they need to. So what causes this is one section of chromosome 7 is deleted, and that's it. This amino acid is deleted, and from there, that leads to cystic fibrosis, just like that. Now, we're going to look a little bit further into chromosomal disorders. We can determine what a disorder is based on a karyotype. So a karyotype is just laying out all of the chromosomes in order, and then you can see if there's multiple or less than what you're looking for, or changes that you aren't really expecting to see. So chromosomal disorders are usually fatal. It's estimated that up to 50% of early miscarriages involve some sort of chromosomal defect. And then if you do come to full term, right, but you have a chromosomal disorder, it usually results in mental and physical abnormalities. So that's just something it's going to be a common theme as we look at the symptoms of various chromosomal disorders. So this is an example of a karyotype. Like I said, it just lays out all of the chromosomes you can see in order 1 through 22, and then your sex chromosomes is your 23rd pair. Females have 2 X's, males have an X and a Y. So again, just normal layout of the chromosomes. So now we're going to look at some karyotypes that show the chromosomal disorder. So one example is Down syndrome or trisomy 21. This occurs in 1 in every 600 to 880 live births. Some of these symptoms are mental retardation, characteristic features, and congenital heart defects. 1 out of 6 die within the first year, and the average lifespan is about 16 years. People who have Down syndrome typically have a lower IQ and really characteristic features like short stature, broad hands, stubby fingers and toes, and a wide rounded face. So I wanted to show you this karyotype, and you can see here that on chromosome 21 there are 3 there. So trisomy means 3, and so when you're looking at a karyotype and you see more than 2 chromosomes and a number, you know that there's a chromosomal disorder just by looking at it. And in this case, because there's 3 on 21, you can see that. Turner syndrome is a sex-linked chromosomal disorder. This is monosomy X. That means you only have one X. It occurs in every 1,500 to 5,000 births. You are biologically considered a woman with Turner syndrome, and you can live relatively normal lives, but you're unable to bear children because you don't have functioning ovaries. And some characteristics of this disorder is a short stature, broad neck, broad chest, no effect on intelligence. And here you can see in the karyotype that there's only one X instead of having 2 X's. And then the last one we're going to look at in this category is Klinefelter. So this is XXY. And when you're dealing with your sex chromosomes, if you have even one Y, you are biologically considered a male. So this disorder is actually relatively common. About 1 in every 500 to 1,000 males is affected. And there's not a ton of symptoms related to this. Considered relatively normal. Characteristics are you're tall and you're sterile. So testes don't form properly, which means that you do not produce viable sperm. And in this case, you can see in the karyotype again that you have an XX and then another Y. And the last thing we're going to talk about is multifactorial disorders. So again, this is disorders caused by multiple factors. You could have a genetic predisposition, like you are predisposed to get this disorder, environmental factors, diet and exercise, lots of similar factors can affect one disorder. One example of this is breast cancer. So a lot of people who develop breast cancer or ovarian cancer don't have a history of this disease in their family, and only 5 to 10% are caused by inherited genetic factors. But those cases that have a genetic factor leading to this result from an inherited mutation in the BRCA1 or BRCA2 gene. Those are tumor suppressor genes. So those genes are mutated and it leads to breast cancer. So most cases develop as a result of multiple factors leading to cell cycle regulating genes. And our last example of a disorder is hypothyroidism. What this means is you have an underactive thyroid. It's a really common condition where the thyroid gland makes too little of the thyroid hormone. So this gland produces the hormone, and that hormone helps the body grow and develop, and it plays a role in metabolism. So about 10 to 20% of the time, the condition is caused by an inherited defect that alters the production of that hormone. And it can be caused by an autoimmune disease that just attacks the thyroid gland. Surgery or radiation to treat thyroid cancer can lead to hypothyroidism, a rare genetic event where a mutation is acquired. But the most common inherited form of hypothyroidism is a defect of the TPO gene, which is found on chromosome 2. And this gene's role is basically thyroid hormone production. So a mutation there is going to influence the production of those hormones. Okay, so now you've got a basic understanding of genetic mutations and what they're caused by and some good examples of genetic disorders. So now let's dive further into your research, into your own choice of genetic disorders.